Hi everyone, and many thanks for the opportunity to present Prescient to you, in particular Omni-CAR, which is a universal immune receptor, creating next generation cell therapies that are controllable, flexible, and adaptable. My name is Yatomi-Clarke, and I'm the CEO of Prescient Therapeutics. Everyone at this forum would be very well aware of the key challenges confronting adoptive cell therapy. They're listed here. Obviously, the time and cost being addressed from many different angles, control and safety, which targets to use exhaustion and escape. The pioneers of CAR-T themselves, University of Pennsylvania, were very well aware of these limitations and deliberately brought together their cell biologists and protein engineers to create a universal immune receptor that we have licensed exclusively and globally, called Omni-CAR. It incorporates a binding system that is being separately developed and licensed by Prescient from Oxford University called SpyTag and SpyCatcher. It creates an automatic and autocatalytic bond, covalent bond, importantly. What's interesting is that this system can modularize any type of cell therapy, regardless of the cell type and regardless of the targeting ligand. In fact, the targeting ligand is basically anything that can bind to a cell. It could be an scFv, but it could also be an antibody or an Affimer or an aptamer or even a small molecule. And the immune cell could be any type of immune cell, whether it's auto, allo, T cell, NK cell, or otherwise. So once again, a true platform. I'm going to just touch on some of the features of Omni-CAR here. Obviously, conventional CAR-T has no ability to control lytic activity post-infusion, and that's obviously a limitation in bringing this therapy to more patients, whereas Omni-CAR has the ability to control activity post-infusion by titrating up the amount of binder that is administered. You get corresponding lytic activity, and you can even cease administration and the therapy switches off. We're aware that some parties out there are incorporating suicide switches in their systems. That terminates the therapy. This does not terminate the therapy. So you can titrate up, but if you cease administration of the binder, the therapy switches off and awaits your command. If the clinician re-administers the binders, the cells are able to re-engage with those binders, reactivate in the presence of antigen, and recommence proliferation. An ability to target multiple antigens sequentially is a unique feature of Omni-CAR using the same cell product. So obviously directing against one, dropping in another binder, and being able to direct that one cell product from one to another. Very important in rapidly mutating malignancies such as AML. If we can do that, we can also address multiple antigens simultaneously, and this is an important, especially important in solid tumors, where we're looking to make the most of the antigens that the heterogeneity of those tumors, and tackle it from all angles. So this has the ability to address not just 2, 3, 4 different binders at once. We're aware that there are other universal immune receptors being developed. We believe this is the first and the only covalent system, and you don't want these things coming off, but as we can see here, a head-to-head, this is the most comparable experiment that this one that Penn did, using the same system and switching out the covalent from non-covalent binding, and you can see that the covalent binding improves the loading of the binder, but also massively improved lytic activity. So not just efficacy, but the other advantages there and a predictability. And from our engagement with regulators, they are comforted by the fact that the binding is covalent and does not come off. What we've demonstrated here is the ability to progressively load each cell with increasing amounts of binder, and when that happens, you get a corresponding increase in lytic activity. So this is I guess a dose response curve that we might be more familiar with seeing with biologics and small molecules, and here we see the same dose response with a cell therapy product in the presence of increasing amounts of binder. This system is quite unique in that it is able to bind many different binders at once. And here, what the team has done is use EGFR, an EGFR binder and a HER2 binder. And you can see here it can bind one or the other or both. And when we do this, it is able to direct at will. So when we load it only with a HER2 binder, it will only kill HER2 positive cells and leave the EGFR positive cells alone, and vice versa. When we load it against arm it with EGFR binders, and when we load it with both, it will kill both, and importantly, will do so just as potently, whether it is dual armed or singly armed. This is using version 1 of Omni-CAR. We are now working on version 3, which is faster and stronger binding, and we've actually demonstrated 3. So we can a very obedient system. We can load up to 3, and we haven't even tested the limits of this. It could be 4 or 5. And it can arm in the proportions that we wish, and by doing so, direct it to kill exactly what we wish. So a very obedient system. And here we can see this is using a HER2 Omni-CAR in an ovarian cancer model. You can see here just the cells alone does not kill any tumor. Once you give the binder, the system switches on, and the more binder you give, the more tumor you kill, and the longer the subjects live. So again, you're harnessing the unparalleled cytotoxicity of a T cell, but with a dose response and a predictability that we would expect with a conventional therapy. This is a recent bit of data that we've demonstrated in a HER2 model, where we have demonstrated that yes, we can get a nice clean cytotoxicity. We can make that killing curve steeper or otherwise according to various levers that we're pulling there. But what we have done is let these cells rest and unarm. So the same cells are unarmed, and by dropping in another binder, there are no new cells, we are able to reactivate and rearm those cells, and you'll see that it is not only the same level of killing, but it is the same kinetics of killing. So whether they're rearmed or pre-armed, it looks to be very predictable, very potent and very predictable, whether they're rearmed or pre-armed. Another bit of data we produced recently was looking at redirection. So in this case, we were using GBM cells, either positive for EGFRB3 or HER2, and putting those both into a well and adding only Omni-CAR cells armed against, initially, EGFRB3. And as we would expect, the HER2 positive cells are left alone. There's no off-target killing of those, and you get rapid cytotoxicity against EGFRB3 positive cells. And then at hour 20, we do nothing but drop in another binder. So there are no new cells. We are dropping in a HER2 binder at this point, and what we see is rapid redirection. So, whoops, just go back. So we see rapid redirection against turning these cells against HER2 positive cells and away from EGFRB3. So once again, rapid switching and rapid cytotoxicity just by dropping in the binder. So showing the unique ability to redirect these cells. Regarding immunogenicity of Omni-CAR, we have done in silico testing, which is over predictive, showing that it has got comparable immunogenicity to humanized and human antibodies. So very low immunogenicity. So where are we going with this? So we're progressing 3 trials in-house, but the bigger picture is a world with a personalized plug-and-play system with Omni-CAR at its heart. So yes, we get tumors sampled and their mutational profile characterized, and we see a world where a clinician will then say, based on the profile of this patient's tumor, we need binders A, B, and E, and taking those off the shelf and plugging them into an Omni-CAR cell and giving a bespoke therapy to a cancer patient. Often we see personalization coming at the expense of efficiency and vice versa, but here quite the opposite. We see an efficient system that is bespoke to a patient and a patient's tumor. There are various manufacturing advantages too with conventional CAR, especially if we're targeting many different antigens. At the moment, we're limited to doing multiple runs, each of them very expensive and time consuming and obviously cost prohibitive, even though we've seen several ISTs now, especially in solid tumours, where we're starting to make a meaningful dent in some of these solid tumours, and a durable one, but it takes many, many infusions. With OmniCAR, we do the hard work once, which is the manufacturing of the cell, and thereafter it is all about the binder, which is not only more cost effective, but it is timely. There is no delay, obviously, and the ability to redirect and create a truly multivalent therapy with the one cell therapy product. I'll finish off by speaking about a summary of our internal programs. We are running 3 programs. The first one is in AML, where we are targeting CD33 and CLL1. The features that we're looking to exploit here to overcome some of the challenges that have faced CAR-T is titrating up, being able to titrate up activity in a fragile patient population, co-arming against 2 pretty important antigens, and doing sequential and simultaneous targeting, and these are validated targets in AML. Another one we're looking at is HER2 positive solid tumours. HER2 is one of the most validated antigens in targets in solid tumours. We need to make better use of it. Obviously, it has some presence in healthy tissues. So again, being able to overcome some of the safety issues there by being able to titrate up and titrate down when we need to. And again, being able to persistently administer the binder as if we were administering several rounds of cells. We also have some proprietary technologies that we are using to address trafficking and overcoming an immunosuppressive tumour microenvironment. Lastly, we've got a GBM program where we're looking at EGFR, V3 and HER2, and another undisclosed target. The same with AML, there's another undisclosed target. Again, being able to co-arm, we've got a very plastic, rapidly mutating disease, and one target's not going to cut it. I think that's broadly appreciated now. Being able to persistently give that binder once again, and being able to switch and provide a multivalent approach is going to be very important in this disease. So just in summary, OmniCAR seeks to address a lot of the problems encountered by current generation CAR therapies. It's the only covalent modular system that is out there, and it is given rise to 3 programs that I've just summarised on the previous page in AML, solid tumours, HER2 positive solid tumours and GBM. We're very open to collaboration and licensing with third parties, whether it be on your binders or your cells or your programs to modularize those, and through modularization, create something that is flexible, controllable and multivalent. So we would love to collaborate with you. We're agnostic on target, we're agnostic on cell type. So please give us a call, and look forward to your questions. Many thanks, and have a great conference.